And speaking of clinical trials, do you see a space where we're going to start to see the ages of enrollment in clinical trials start to reduce, thinking through that, risk perspective and the concept that change is already happening in the brain, 20 years often before symptoms are developing? Do you think we're going to start to see clinical trials that are going to be looking at individuals earlier and following them or in a longitudinal pathway?
This is one where I think it's sort of this trade-off, and we've talked about this a lot in the Alzheimer's space in terms of study design, that you can either study the disease when it's measurable or when it's changeable. Can you intervene early enough to make a difference and late enough that you can measure that difference? And for proof of concept studies that are really small sample sizes, it's very hard to do both. If you're a big company and you're in phase 3, you can just put more patients into the study and you can measure really slow, subtle, early change. But if you're a small company and you're trying to get proof of concept, you need to measure patients that have enough decline. And sometimes when you go to those earlier, younger patient populations, the decline isn't quite as steep and you're not able to see the treatment effects that you need to. And so there will always be a little bit of a disconnect between the population you need to study in a proof of concept small trial and then the group you would study in a phase 3 that might be kind of intermediate, and then the group that will eventually be treated out in the community. And it's important to make those decisions carefully because otherwise you can miss proof of concept when you truly have a treatment that works. So you don't want to try to go too broad too soon, or you might miss your chance for success.
Understood. Jeff, do you have anything to add to that?
I would just add that an important part of this dialogue is about biomarkers and their regulatory status,
Yeah.
so that a trial could have a positive outcome of a delay in biomarker maturation if the regulatory agency had enough confidence that that biomarker would eventually predict clinical benefit. And we're in the process now of accruing this kind of information which would allow us to have that kind of confidence. So I think there are a lot of moving pieces here, but two of them are the regulatory part of this and the biomarker part of it.
Yeah,
Understood.
one thing I want to add to that, the biomarker part is interesting because at first you can have a biomarker that could be an accelerated approval biomarker where you get full approval, but it's accelerated approval. You still need to confirm it with a clinical trial based on, so you do the study based on the biomarker, you confirm it with a clinical trial that then shows a clinical benefit. But the second option is really the direction that we should be trying to go, and we obviously are trying to go, which is where you have a biomarker that's established well enough as a surrogate that you get full approval without any kind of conditions. And it's not accelerated approval, but it's actually full approval based on a fully established surrogate. And so getting our biomarkers to the point of surrogacy is really critical because if we can get full approval using biomarkers, that allows us to be studying patients early. It allows us to be in the stage where we're making the biggest difference and see the biggest benefit. And so that is the direction that we want to be going. And maybe an intermediate step is having a biomarker that could be accelerated approval outcome, and then moving it to where it could be a standalone surrogate and be able to be full approval, have full approval based on that outcome.